Okay, everyone, we're going to get started now. I just want to say hello, and my name is Renaissance McIntyre. I work with the Master of Science in Biomedical Informatics program. And I just want to thank everyone for joining us for our monthly seminar series lecture. This month, we are proud to have Dr. Bowden Homechuk do a presentation for us. He works for the Department of Medicine at the University of Chicago. His research field is cardioinformatics, and his focus is on creating biomedical informatics infrastructure platforms that provide powerful query and browsing interfaces between data and user. To that end, Dr. Homechuk is the lead developer and maintainer of HeartBio Portal, which is the world's largest human cardiovascular and cerebrovascular genetics data ecosystem. So now I'm going to turn it over to Dr. Homechuk to discuss his research and HeartBio Portal. But I do want to mention before we get started that if you do have any questions during the presentation, please type them in the chat box and we will get to them at the end. All right, I'm turning it over to you now.
Thank you very much, Renaissance. Appreciate it. Hi, everyone. Nice to see such a nice turnout. Glad to give this talk to you guys today. Really excited about it. So I wanted to just jump right in. Thank you very much, Renaissance, for the nice introduction. I'm an instructor in the Department of Medicine. I work with Andrei Rosetsky and Bob Grossman, also in the Department of Medicine. And we develop some really cool tools for facilitating biomedical informatics research. So with that being said, let me just go ahead and kick off. The title of this talk is Cardioinformatics and its impact on healthcare. Basically, we're going to be discussing what it's like to perform computational research for the cardiovascular disease research community. And we'll discuss a number of different things. So first thing I wanted to discuss was career trajectory. So I was asked to actually talk a little bit about how I got into the space. And basically like what my career looked like. So I split this up into different phases. So the first phase was in college, I triple majored. I did a bunch of STEM stuff. So I did molecular biology, biochemistry, I did math, I did physics. Ultimately, the end result of that was I had fun trying to balance coursework with NCAA college golf. I played for the for my college golf team. Obviously, I think a little bit, I was too overambitious, so I had to drop out my junior year, unsurprisingly. But in the end, like I acquired pretty decent preliminary skills and technical training to conduct quantitative and computational biology research as part of my college experience. And plus, I had a number of different summer internships, including international ones like at EPFL in Switzerland and other places. So that's kind of phase one of my career. Phase two of my career was I went to graduate school. I did a PhD in human genetics and genomics with a focus on bioinformatics. So they provided us with two tracks, one being the computational track where you take a bunch of courses similar to the kinds of courses that are being taken in your Masters of Science Biomedical Informatics program. And then, I also supplemented with programs in stats and other fields. My PhD was composed of a pretty wide, eclectic mix of a number of things. So I built a number of different front-end and back-end infrastructures that ranged from ChIP-seq and RNA-seq bioinformatics analysis pipelines, high-performance computing applications, specifically for heat map visualizations for big data. Served as a bioinformatician collaborator on several computational epigenetic studies. So that was a lot of fun too. I got a chance to work collaboratively across a number of different large scale studies that were being conducted where I basically served as the main bioinformatician who did all the analysis. That included also my first sort of experience working with cardiovascular disease. Actually, my interest in cardiovascular disease as a research field from a computational point of view started around then. So this was like my, probably around my second year of grad school, which is when I got really excited about it. I also co-authored two chemistry textbooks published in CRC Press, Taylor and Francis, due to my interest in teaching and drug discovery. And then I even got married and had two kids. So the end result was I almost lost my sanity, but, I went to Stanford afterwards. So, I was hoping to get it back a little bit. So I went to do my postdoc there. So that brings us to phase three. So I did my postdoc at Stanford with a focus on bioinformatics and computational biology. I was in 2 departments at the same time. I was in the Department of Biology and the Department of Medicine, specifically the Division of Cardiovascular Medicine. I actually ended up doing sort of like 2 postdocs in one. I worked for 2 postdoctoral advisors simultaneously, so Orgozani in biology and Tim Asimas in medicine. Don't know if I would suggest that for everyone. It's sort of like you do 1 postdoc, but it's double the work. But I was really fortunate to get a postdoctoral fellowship from the American Heart Association to support my work there. So that was super exciting. I also got involved in the NHLBI TopMed initiative. I was specifically working in the atherosclerosis working group. I also served as a research associate in the VA Palo Alto healthcare system, working on cardiovascular disease genomics in Korean and Vietnam War veterans that were enrolled in the Million Veteran Program. At that time, we had about 450,000 whole genome sequenced. Now it's way closer to the million mark. But at that time, the program was, just kind of getting started, and it was a really fascinating experience. I had a walk appointment. Walk means work without compensation, so I couldn't double my salary. Like when you work as a postdoc, you can't take salary from another place. So although I was a research associate, I wasn't getting compensated for it, but it gave me some really nice experience working on this kind of data. And this was as part of my postdoc with Tim Asimas in medicine. He is one of the co-PIs of the Million Veteran Program. So this is how I got excited about it. During my postdoc at Stanford, I built and published HeartBioPortal, which is currently the world's largest cardiovascular disease genetics data ecosystem, as Renaissance mentioned. At the time, it was not, but I've been working on it pretty diligently ever since. And sort of the vision is to create the one-stop shop premier place for hosting all of the world's heart disease and stroke data. So we've been making some really good progress on that. End result, again, almost lost my sanity, but won the national search for a faculty position at UChicago, which is, where I am now. Phase 4, so this is where I am now. So I'm on the Pathways to Independence track. I'm working with Andrei Rzetsky and Bob Grossman in the Department of Medicine, specifically the section of computational biomedicine and biomedical data science. My principal research focus is, as Renaissance mentioned, creating essentially powerful querying and browsing interfaces between data and user. So in this specific context of this talk, the focus is on HeartBioPortal and what you can do as a user to query and search for various cardiovascular and cerebrovascular disease genetics data inside of HeartBioPortal. Basically, what I'm doing is I'm continuing to develop and maintain HeartBioPortal, which enables gene and variant level search across various heart disease and stroke phenotypes for the broader disease research community, specifically with a focus on cardio. And I work directly with Bob and Andre, and these are pioneers of large-scale data commons, and Andre is a pioneer in the use of bio-NLP methods and various large-scale mathematical modeling and various other techniques to mine big data. So this is where I come from. So put together end result of this, well, we'll see, right? To be determined. No one really knows where things will go from here in terms of my career, but we'll keep aiming for the tenure track. So I am currently an instructor of medicine. We'll be applying for tenure track assistant professor positions probably sometime next year, once the hiring freeze is all clear. A lot of places have hiring freezes until the summer of 2021. So research mission of my lab, sort of build a world-class research program in the emerging field of cardioinformatics, working at the nexus of bioinformatics and precision cardiology. Also perform service and public outreach and actively pioneering and promoting cardioinformatics to the scientific community. My goal is to try to fight heart disease with computation through the creation of large-scale cardiovascular disease data commons technology, specifically exemplified by HeartBioPortal. I'm also a big advocate for the need to not only bridge the bench to bedside divide, but also the informatics to medicine divide in cardiovascular healthcare. I think not enough people talk about that. This is something that one of my mentors, Bob Grossman, talks about a lot, and I feel like more people should be aware of it. An example of fighting heart disease with computation, a lot of people ask me, what does that mean exactly? How do you do that? What does that actually translate to? So one example is using data-driven cardiometabolic drug discovery. So essentially trying to use omics data, for example, within HeartBioPortal, to perform systematic cardiovascular disease target identification and prioritization of various drug targets and biomarkers. The ultimate goal, of course, is to try to impact healthcare and with it, genetic medicine. And then maybe just one day we'll see another AHA journal, maybe like Circulation Cardioinformatics or something, to really, bring the community together towards publishing this kind of work and sort of trying to fight heart disease with computation. I think that'll be really important as well. So I mentioned cardioinformatics a lot. So cardioinformatics, it's a published pretty large review article. It's got like 250 citations. It was really exciting to write. It's something that just came out in December of last year. There's also an associated press release, if you want to check that out, sort of summarizing the main sort of punchlines, of which there's a number of different punchlines. We'll cover them later in this talk. Just wanted to highlight a brief quote here by our section chief of cardiology. It reads, "The field of cardiovascular disease has always been data and technology driven, but when it comes to taking advantage of the vast amount of information that already exists and developing a systematic approach for analyzing it, the field has fallen short. The emergence of computational biology and bioinformatics in the cardiovascular field will provide insights and potential therapeutic avenues for the treatment of cardiovascular patients." So you can clearly see that there's a critical unmet need here, and basically the job of my lab is to try to close that gap. Also, when you look at the Cardioinformatics review article published in Briefings in Bioinformatics, I also made all of the data, source code, and analyses publicly available through GitHub and also archived through Zenodo. So this has a DOI and everything, so you can actually cite the data, you can cite the analyses, you can cite the actual source code if you ever wanted to take the source code and build off of it. So if you wanted to look at various research questions of interest to you using our source code and the analysis that we generated, like all of this is available to you as the user. So that's pretty cool for reproducibility purposes to power the kinds of visualizations that you'll see in the next few slides. So without further ado, let's look at these few slides. So globally, over 17 million people die from cardiovascular disease per year, making it the leading cause of death worldwide. An even more crazy statistic that people, when they hear it, they're like, wow, that's really incredible, is that by the year 2030, so 10 years from now, almost half of the adult population in the world will have some kind of cardiovascular disease diagnosis. So that could range from something mild like hypertension, or perhaps something more intermediate like coronary artery disease, or perhaps something clinically fatal like a heart attack or a stroke. But the point is that by a decade from now, half of the adult population will have a CVD diagnosis of some kind, which is pretty scary. So the motivation is that we're dealing with the #1 global killer. Mortality rates in the United States are basically the same as in the world, right? Also #1. And we kind of want to do something about that, and we want to fight heart disease with computation to see what can actually happen. This is actually a theme I want to bring up over and over again in the talk. When we think about cancer, right, and we think about precision oncology, we know that, computation has had a huge influence, right, on the way we conduct clinical care for cancer patients. And essentially what I'm advocating for is that we do a similar thing for cardiology. Can we also use computation to improve the translation of clinical care, the informatics to medicine divide that I brought up on the last slide? That's basically a key. Like if you take only one thing from this talk, right, I want you to take that away. Like that's the take home message, is can we do what precision oncology did, but for cardiology, for heart disease, for strokes, etc. The thing is, to do something, you need money, right? So CVD research is underfunded, at least relative to cancer research. So I ran the calculations for this, and like I mentioned, like all this code and analysis and stuff is available online on GitHub and indexed through Zenodo, so you can rerun these analyses yourself for the latest data. So this is not taking into account like late 2019, early 2020 data. But basically, the take-home message is that NIH funding for cancer versus cardiovascular disease since the year 2008 has had on average $500 million, so $500 million less per year, which is a pretty large amount. This categorization was done by the NIH starting in the year 2008 because that was the year when NIH implemented the system called the Research Condition and Disease Categorization System. It's basically a way to index all of their sort of internal funding data. So that's why there's no data before 2008. Here, we downloaded data from the NIH Research Portfolio online reporting tool. So that's the report tool. You may have heard of that if you ever wanted to look up someone's grant. So if you Google a PI's, not Google, if you search for a PI's last name and in the report system, you'll see this information. And the way we classified cancer versus cardiovascular disease was, for cancer, we basically calculated it from projects categorized as, for example, brain cancer, breast cancer, cancer genomics, cervical cancer, et cetera, et cetera, et cetera. There's like a number of different categories we used. And then for cardiovascular disease, we looked at anything with the word cardiovascular, cerebrovascular, congenital heart disease, hypertension, pediatric cardiomyopathy, stroke, et cetera. So that's how we performed this calculation. Here we're looking at primary research articles, excluding review articles. So we're text mining PubMed. So we found that when you compare cancer papers versus cardiovascular disease papers that use bioinformatics methods, there's also quite a huge disparity. So in addition to the funding disparity that I showed you on the last slide, there's also a publication disparity. That's not surprising, of course, since publications are funded by federal tax dollars via federal grants, right? So if there's a funding disparity, there's also going to be a publication disparity. So clearly, you see here that there's a small computational footprint in cardiovascular disease relative to cancer. And the way we analyzed this was we basically, as I mentioned, we text mined PubMed. We looked at medical subject heading terms, AKA mesh terms. And we looked at mesh terms in PubMed that were tagged for things like bioinformatics and genomics. And since all of the queries are based on manual mesh catalogs, basically the 2018 tallies here, the small thing that you see, they'll obviously lag behind the current volume of publications. So for the most up-to-date results charts, you have to rerun the code. So you have to go to the GitHub repository behind a review article and actually rerun this code. So the pattern will stay the same. Next, if you look at the bioinformatics footprint in cardiovascular disease, it's actually small in general, not just relative to cancer. So if you plot out all the cardiovascular disease papers that are coming out, you see there's a lot of them, right? There's way more cardiovascular disease papers, for example, being published than papers being published in all of bioinformatics. So regardless of field. So it's a very, 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 like vibrant research space, right? There's a lot of CVD papers being published, but of that, there's only a very small percentage of cardioinformatics papers that are being released. So those papers do not include bioinformatics techniques or methodologies as part of their study design, which is pretty crazy. So again, we're using all mesh terms for bioinformatics and genomics to take a look at this. Likewise, there are far more CVD papers released annually per year, as I mentioned, than bioinformatics papers in any domain area, not just necessarily in CVD, which highlights, I think, the tremendous momentum and volume of new research results that are being published in the cardiovascular disease field, which is actually quite fascinating how huge that momentum is. So basically the take-home message here is that we need more bioinformatics in cardiovascular disease, and of course more funding for it, since the funding is what gives rise to the research, gives rise to the publications. So it's a question of changing the culture. So we know for a fact that CVD is the #1 global killer, right? We know that it causes over 17 million deaths per year. We also know, as I mentioned, that by 2030, almost half of the adult population will have a cardiovascular diagnosis of some kind. We also saw that there's an NIH funding disparity that exists in CVD versus cancer research. And funding for cancer research has been, on average, as I mentioned, $500 million higher than that for CVD since the year 2014. Bioinformatics footprint in CVD is modest relative to cancer, and my hypothesis is that if you expand this footprint with improved data science, so bioinformatics techniques, this will ultimately improve healthcare, as has been the case for precision oncology. So this is sort of, I mentioned, if there's one thing, if there's one theme that you take out of this talk, it's that can we do for cardiology what computation has done for oncology? Can we, push the cardioinformatics needle to ultimately impact cardiovascular healthcare? So let's say, assuming that cardioinformatics can potentially impact healthcare, where would we start? So this is the question that I asked myself many, many years ago when I was first contemplating, like, where can I, as an individual contributor, try to make the biggest difference? And for me, the answer to that question started with the users. I wanted to start with users because, you know, doing a postdoc, you know, at Stanford, I think, you know, the whole Stanford environment, right, is all about, you know, users and value and how many, you know, like people use your software, use your platform, right? Like the whole, you know, Facebook 2 billion users thing. I mean, obviously we're not talking about Facebook here. You know, we're talking about cardiovascular genetics, right? I mean, obviously that's going to be a much smaller, you know, portion of the global population. But the point is, can we build something people want? So the objective was building something people want that targets a really concrete, specific, you know, user base. In our case, we wanted to capture the entire broader cardiovascular disease genetics research community, which is a relatively small but a rapidly growing user base. So when I first set out on this path, first thing I did was I looked at cancer. So I said, which cancer resources have many users? And right now and back then, a few of them came up. So the two major players are the Genomic Data Commons, which is what Bob Grossman is the main PI of. So really valuable to get a chance to work with him on this. You know, back when I was doing this as a postdoc at Stanford, I had no idea I would be working with Bob someday on Heart Bioportal, which is pretty cool how, you know, like our fates sort of, you know, like came together. And then there's also cBioportal, which is something that's been around for a little bit longer. But you see, these are publications, highly cited publications that have been cited hundreds, thousands of times. And I wanted to make something like this for the cardiovascular disease genetics research community. And I wanted to provide, you know, most importantly, not just a publication that's highly cited, but a platform that can provide intuitive visualization and analysis of large-scale data sets for the broader research community. So we wanted to follow, the same model that cBioportal, for instance, followed, which is why we actually named our platform Heart Bioportal. It's sort of a hat tip to the original cBioportal vision that, you know, you can create something like this for cancer. So I wanted to do something like this for cardiovascular disease. And these two resources, right, GDC and cBioportal, they capture effectively 100% of the market share of cancer genomics researchers. So we wanted to do the same thing for heart genomics researchers. Ultimately, we ended up publishing a paper on this. So this was the first paper, the first publication that appeared in April of 2019. It was a paper published in Circulation Genomic and Precision Medicine, which is like the top circulation journal for computational research. Remember a couple of slides ago, I showed that we should have, you know, sort of a sister journal called Circulation Cardioinformatics. I mean, this is basically the equivalent of Circulation Cardioinformatics, right? So this journal, you know, people refer to it as Circulation GPM. GPM is basically like, you know, it's sort of the venue right now for the American Heart Association community to publish computational work in the field of cardiovascular and cerebrovascular disease. So this was a paper that I co-authored in April of last year with my two postdoc advisors, Orgozani and Timasimas here as senior authors. Heart Bioportal, you can access it through just, you know, heartbioportal.com. It's a secure interface. Basically, it looks pretty much sort of kind of like Google, right? You can search for specific gene names, and there's a lot of data behind this, right? So here it says, you know, there's almost 45,000 human genic and intergenic regions across almost 7 million variants across 17 cardiovascular disease phenotypes and 56 cardiometabolic quantitative traits. I mean, there's a lot of data in there. So currently, you know, we're now the largest cardiovascular disease genetics data ecosystem in the world, and we also have a Twitter account. So here's our Twitter account. By the way, speaking of Twitter, I'm super active on Twitter. I really like Twitter. So bottom right-hand corner of like all these slides has like my Twitter handle at Bodenome. So you know, if you guys want to like follow, you know, the kinds of, you know, cool cardiovascular disease computational research that's been coming out, over, basically on any given day. I'm super active with trying to keep up with what's happening in the cardioinformatics space and try to tweet out about it as much as possible. So Heart Bioportal also has its own Twitter account. It's, at Heart Bioportal. And as you can see, it's a data commons for browsing, searching, and analysis of human genetic information related to heart disease and stroke. So when I think about, how am I contributing to cardioinformatics, right? So this is a whole area, right? And there's many different sort of niches in this area. My specific niche is, personally, I write scalable and secure engineering infrastructure to power the world's cardiovascular disease genetics genomics data, right? I don't produce data. I collect data and I analyze data, and then I make that data accessible to the broader research community. And I try to then leverage that data to come up with new insights into a number of different areas, which I'll be happy to cover in the supplementary slides to this talk. But one of the most important questions, I think, that, as an MSc student in the biomedical informatics program is, if you're interested in cardioinformatics as a field, ask yourself, how can you contribute to cardioinformatics? So to answer this question, there's a number of ongoing areas of research, many of which we cover in full within our cardioinformatics paper, which I encourage you to read for a full description on each of these topics. But you see, there's a number of different topics that you can get involved in that concern fighting heart disease with computation. My specific area is building data commons. Your specific area might be something like cardiovascular natural language processing, for instance. So I only highlight that during this talk because actually it's interesting that, how very often when you read a paper, you want to go to the data availability section and you want to pull down, the data that was used to power the, the analyses of what you're reading. And, sometimes there's no data availability section at all. Sometimes it says, data available upon reasonable request. And then, you email the, the corresponding author, and you either never hear back or, you usually never hear back. So this is actually a huge problem in cardiology. I think way more so than in other fields. So thankfully in oncology, most papers and a lot of journals mandate, the release of data as part of your publication, but that hasn't really reached the cardiovascular disease research community yet. So a lot of the findings that you read about in the literature are actually, they don't release their data, which actually makes that data sort of invisible to a place like Heart Bioportal. I like to call it like dark matter, right? Dark matter data, like data that, you can't actually mine or do anything reproducible with, and you can't actually search it within Heart Bioportal. And the only way to really search for it would be to perform some kind of sophisticated, NLP text mining on the original publications. So there's like a huge area right now, like that's just now emerging into trying to do cardio NLP research, or like bio NLP research, but specialized for cardiovascular disease. It's an area I'm very interested in, an area that I'm writing grants in, that I'm really excited about. So, if there's any interest, in that, I encourage you to also reach out to me personally and ask about, opportunities to work with me on stuff, or just in general guidance on, if you're interested in cardioinformatics, like as a first-time researcher, as a first-time user, like where would you start? I'm happy to give advice on any of the above. So just to wrap everything up, I know this was supposed to be a 30-minute talk, so I'm coming up to 32 minutes here. I wanted to acknowledge some recent funding that I received from the Center for Translational Data Science Pilot Award. Super excited about that. Tweeted out about it on March 9th. And then just want to open up the floor for Q&A. And if anyone wants to ask me questions about, Heart Bioportal itself, sort of what it does, I have a huge amount of supplementary slides here and screenshots of all kinds of new and existing Heart Bioportal features, including features that will appear. So things that I'm currently working on, for example, in cardiometabolic drug discovery, sort of turning Heart Bioportal into a drug discovery dashboard. This is something that's also in some of my supplementary slides here. So if you want me to, run through some of that with you here on the call, on the Zoom, here on Zoom, or if you want to discuss, sort of my outlook on any of the things that I previously mentioned, please go ahead and ask away. Thank you. Oh yeah, and please go ahead and type your questions in the chat box if you have them. Or you can unmute your microphone and just go ahead and ask. All right, so the first question I see here is Megan Puckleworth from Northwestern University with Beth McNally talked about performing molecular autopsies on sudden death patients, so I assume sudden cardiac death, but none of that is ever published. Do you think you can ever capture these kinds of institutional findings? That's a good question. So yes, I can. Actually, this is something that I'm currently doing with Bob Grossman. So currently, if you use HeartBio Portal, right, you basically, you can type stuff in, you can find stuff, you can download it, and try to understand, you know, like what is currently publicly available. However, if you wanted to see controlled access data, right? So if you wanted to see things in dbGaP, for example, or you wanted to see things in TopMed, for example, et cetera, you have to obtain permissions to see that data. And unfortunately, without you know, applying, you know, for IRB approval, you can't see, for example, what is currently known from a gene level, variant level perspective in controlled access data sets like the TopMed cohort, for instance. So with Bob, what I'm currently building is I'm trying to build sort of engineering infrastructure that allows you to log in and have the system recognize whether or not you've already received approval for viewing of specific controlled access data. And if you have approval, to show that data within the HeartBio Portal interface. So to actually display it in the UI and to provide it in downloadable form, so you can actually download, for example, the summary statistics. So I like to think of this from you know, let me think about a good analogy here. Think of it like, you know, instead of building up, like if you're building a building, right? Instead of building infrastructure, you know, to build a taller building, what we're actually doing is we're building underground infrastructure. So we're building down, not up. And when I say building down, we're trying to basically make controlled access data, such as, for example, the one that you mentioned at Northwestern University, make it accessible to users that have prior permissions. So in that sense, for example, if you're a Northwestern researcher and you already have permissions to, as a Northwestern researcher, see some of this data that's pre-publication stage, and you wanted to analyze it relative to everything that's known about sudden cardiac arrest in the publicly available data sets, and you wanted to sort of, you know, harmonize and compare your data set against the global data sets, you'll actually be able to do that. So that's sort of the vision behind how we're expanding HeartBio Portal now, is we're basically building down instead of up. We're trying to open up those controlled access hidden data sets to users that already have prior permissions, or for users that don't have prior permissions, but they want to apply for it, we're streamlining that data access request process. This is actually something that's being worked on very heavily right now by many different resources at NCBI. Because the dbGaP submission process is pretty clanky. So there's a number of different resources coming up now, like, for example, the NHLBI data stage program that is streamlining the entire data access request pipeline process. So this is something that we're actually directly involved in as well as developers and maintainers of HeartBio Portal. Anyway, very long-winded way of saying, yes, we can eventually get those kinds of institutional findings. We just need to build the computational infrastructure to support it. Next question. Is HIPAA an obstacle to data collection? So again, so this goes back to my to the previous question. It's all about, do you have IRB approval to view that specific data? So if you build the infrastructure to support users who already have permissions to see data, that will step around that obstacle. Next question: how specific what you do to cardiology? So with respect to cardiology, so how specific what you do to cardiology? So this is, you know, all the data that we currently have in Heart Bioportal is all cardiology focused, mainly on cardiovascular and cerebrovascular disease. At the same time, we also recently added support for cardiometabolic traits. So for instance, you can search not just disease phenotype, like for example, hypertension, but you can search also systolic or diastolic blood pressure traits. I'll actually show you a screenshot of that. Yeah, so here's a new, oops, sorry. It's going a little slow. There we go. So here's a, oops. Sorry, my computer is like getting all weird. Let me go ahead and just go to the slide manually like this. There we go. Okay, so here's a new feature that we pushed on April 24th. So if you're interested in, for example, the gene ADD1, which has been associated with salt-sensitive essential hypertension, you can actually see the shared genetic architecture of that gene's variants across the continuous cardiometabolic spectrum from health to disease. So in this specific case, you're looking at all the genetic variants from GWAS studies that are available for diastolic blood pressure and hypertension. And you can look at that intersection here. So when I say cardiology, I mean both disease phenotypes as well as cardiometabolic quantitative traits like diastolic blood pressure, systolic blood pressure, ECG traits, et cetera, et cetera, et cetera. Next question. Thank you for an amazing talk. So if you already mentioned this, but do you see a role for Heart Bioportal in pharmacogenomics and cardiology? Yes, yes. Very happy to see that question because now I can show more slides. Let's see, some good supplementary slides to show this off. Oh, by the way, as I mentioned, the previous question, right? When I say cardiometabolic quantitative traits, there's a number of them, right? So there's like blood cell traits, there's lipid traits, there's kidney function traits, et cetera. Anyway, to answer about the pharmacogenomics. Yes, so here is what the drug discovery suite feature of cardiovascular, of Heart Bioportal will be. So as I mentioned, my vision is to sort of turn Heart Bioportal into a drug discovery dashboard. And part of that vision will be to let users search for a specific gene like TNNT2 and see, for example, what clinical phase of development that gene is currently at when you mine, like, for example, clinicaltrials.gov. And see what is known for that gene across all the different existing cardiovascular disease trials. Also looking at what is the top drug hit currently for that specific drug target, so for that specific gene, including information about that drug. So what type is it? What clinical indication is it designed to address? What is its mechanism of action, et cetera? There will also be toxicity and efficacy information. So is there toxicity in rats? Is there liver injury reported? Does it cross the blood-brain barrier? Is it carcinogenic? Its bioavailability, et cetera, et cetera. And then if you click on this list all drugs section, you will be able to see, for example, you know, a ranked list of number 1, number 2, number 3, including pharmacodynamics information here on the right-hand side, absorption information, and protein binding information. So sort of the objective is to, you know, serve as a next generation infrastructure for cardiovascular disease data worldwide, and ultimately to publish Heart Bioportal 2.0. So as we pivot Heart Bioportal into a drug discovery dashboard, we also want to show how we can use that dashboard to come up with better targets in real time. So we're actually planning on submitting this next paper to either Science Translational Medicine or to Nucleic Acids Research database server issue. And all of it is very focused on pharmacogenomics and cardiology. Thank you for that question. Next question. There are different mutations in different kinds and stage of cancer that makes cancer data size big and useful. Do you think bioinformatics has the potential to help cardiovascular disease as much as cancer? Is there any precision medicine example for cardiovascular disease? So there's a number of questions here. So, okay, so where should I start? Yes. So when it comes to, what kind of a potential does bioinformatics have, to help cardiovascular disease? So this actually gets to the heart of the question of, how hard is it, right? It's an interesting idea in theory. Can we actually do it in practice? So CVD is a lot more complicated than cancer, to the point where, like, you're talking about something that's systemic, right? So you're talking about something that, like with cancer, if you have a tumor, you either do or don't have a tumor. There are pre-malignant states, but at the end of the day, if you get a tumor, you can biopsy it, you can sequence it, you can cut it out, you can put it through an Illumina machine, and you can sequence the genome. With hearts, you can't actually do that, right? So you can't actually cut out a person's heart. At least, if that person is still alive, like they're not going to consent to that, obviously, right? So you can't actually do that. So you have to find other ways to get at the question. So this is why data integration, multi-omics, bringing together data sets from a number of different heterogeneous data modalities is so important. Like, to answer the same question that for cancer would be easy was actually much, much harder for cardiovascular disease, because you have to find a way to triangulate, I think that's the best word here, to triangulate those insights across a number of different data modalities to get at that question. So are there any precision medicine examples for cardiovascular disease? Oh, yes, of course. Yeah. So we talked a lot about them in the Cardioinformatics review article. So I would just suggest the OP to take a look at the paper for those examples. Next question: how accurate are your drug predictions using bioinformatics with actual experiments to find drug hits? So this is actually something that we will find out once we've assembled the data sets. So right now we're organizing, collating, cleaning, harmonizing all of the publicly available cardiovascular disease drug discovery information out there from a number of different resources, ranging from Drug Bank to CHM-EMBL, to open targets, et cetera, et cetera. So once we've brought all that data together, the next step will be, how can we not only make that data accessible to the user, but how can we leverage all that data to create predictive models? Predictive models that could then find better targets, or, for example, show that you can now build a machine learning or AI model to predict already existing targets. So sort of retrospectively see whether or not you can build an AI model on the existing data to predict something that you didn't know before. So that's something that's currently an ongoing area of research in my lab. Let's see, next question. Have you tried mining Nomad or Alpha to predict disease frequencies across populations? Yes, that is something that we mine a lot. So again, going to my supplementary slides here, we provide integration with Nomad, where, for example, you can search for individual mutations. So if you know the accession identifier of your mutation, so let's say you're interested in the gene PCSK9, and you're interested in a very specific variant in PCSK9, and you want to see how that variant is distributed across the world's populations, you can do that. So there's an ancestry chart on the left where you can basically see the distribution of rare and common variants across everything that's known about that gene in that disease. Note that you can set your minor allele frequency. So, for example, if you want to distinguish a rare variant as something that has a math value of less than 0.01, go for it. If you want to have it be 0.05, you can do that as well. You just have to drag the slider bar to see the interactive display of how rare and common variants are distributed across the different population groups. And then if you search for the variant in the global map, you don't see it here, but it will actually pop up these circles that'll show you the allele frequencies of that variant across the different populations. So yes, we provide direct integration with Nomad. Originally, we were doing 1,000 genomes, but then once Nomad caught up with a lot of its data, we started integrating with Nomad. So that's also a part of Heart Bio Portal. And this appears to be the last question that is on my list. So I realize this, I may not have perfectly answered every question. So for anyone who's already asked a question, they want to ask a follow-up question, just go ahead and post here if you feel like I didn't answer your question completely or in full, or if you have a follow-up question to the question that you asked, please go ahead, just type it in here, or unmute your microphone and ask through the mic.
Thank you so much for all of this. While we're waiting for questions, were there any more of your supplemental slides that you wanted to go through?
Yeah, I mean, I'm really happy with with the questions that you guys asked me. So let me actually, given the quality of the questions I got, let me actually go ahead and run through just briefly some of these slides, because I feel like I got a lot of really great technical questions. I almost feel like I should have maybe put a lot of this material into the main talk and not in supplement, but 30-minute talk, so I can't really get to everything. But let me just go ahead and run through this real quick. So Heart Bioportal provides users with the ability to look at RNA-seq and microarray data. So we have a differential expression feature where you can see, whether or not your gene is up or down regulated across a number of different studies. And then if you wanted to pull down additional information, so like the fold change, of that up or down regulation, or you wanted to pull down, the p-value or the FDR value, you could do that too. So just press the download button and you'll see that content. There's obviously a GWAS feature here where you can look at variation type, clinical significance. You can look at the most severe consequences of what you're dealing with. There is, as I mentioned here, the ancestry and variant geography map. There's a really cool variant viewer chart where you can actually look at variants and also their effect on splice isoforms. So seeing, like, for example, like if you have a specific mutation, like a specific missense variant that's leading to a specific amino acid change at a specific position, and the specific phenotype it's involved in, it's all there, so you can look at that as well. These lollipop plots are really, really cool to build. This is actually one of the programmatically was the hardest to build, because this was all JavaScript. By the way, all the visualizations in Heart Bioportal, they're all written from scratch by me in JS. So there's no existing how to say, there's no dependencies on external libraries. So everything here was written basically from, using JS from scratch. If you're interested in JS development, drop me a line. I generally find that people in bioinformatics, computational biology are usually Python and R users, but given how important data visualization is, I feel like JavaScript is a huge, huge, huge place in this kind of work. So if any of you are interested in that sort of a thing, just drop me a line. Also, there's an FAQ page which details, where is our data coming from? So what biological databases are we getting? Where are we sourcing and syncing our data from? So like Ensemble, ClinVar, GWAS Catalog, GWAS Atlas, OMIM, et cetera, et cetera. Also, how we perform the analysis. So how does Heart Bioportal present GWAS results? So all the technical details that you ever wanted to know behind, our bioinformatics analytics pipeline. Also, some new features, so we can search multiple phenotypes for shared genetic architecture. I mentioned this. This was a relatively recent feature where not only can a user look at disease phenotypes, but they can also look at cardiometabolic quantitative traits, and then they can set the stringency of what they want to see. So they can look at, in addition to all these cardiometabolic traits, they can look at, for example, like if you wanted to look at only the significant genome-wide signals, you'd have to set this to 5e⁻⁸, right? That's the usual GWAS filter. So by default, it's set at any p-value, not necessarily just showing genome-wide statistical significance, but you can set this yourself. This we already went through. This is one of the coolest new features that I just recently deployed. Yeah, this we already went through. Then new upcoming features, we talked about, some of the drug discovery work that's happening on the back end right now. We talked about this. And this kind of, the details, how are we actually getting all this data, right? So how are we streaming it? This is a really technical portion of the talk. I can actually give a talk just on this. And I'd prefer to essentially focus on the Heart Bioportal, data user standpoint. But if you wanted to really get into, how are we getting the data? We're basically we're basically pinging endpoints, right? So we want to see, like how endpoints move, how we can track that movement, how we can, for example, if I wake up tomorrow morning and I get an email notification in my inbox saying, hey, the data that you used to pull down from GWAS catalog through their REST API, that endpoint has moved. And then I have to go in and I have to recode, that specific portion of the code base so that on the next update, I don't lose data that's accidentally moved, right? So basically the idea is I want to listen to endpoints and get pinged when they move in real time. So I want to sort of do like continuous integration with the data sources that I'm pulling down from. And that's accomplished through a bunch of async streaming of their REST API endpoints concurrently. So a lot of parallel programming from multiple different set of biological databases. I like to call this the Internet-of-omics algorithms. Sort of like, think of it like IoT, like the Internet of Things, but you're abstracting the concept of things from hardware to software. So instead of something tangible like a device, like your phone talking to your coffee machine to make you a coffee, you're talking about databases sort of talking to each other. So you're establishing this kind of massive integrated e-connectivity of omics data sets across multiple different biological data silos. This was a concept that my postdoc advisors thought was actually patentable. So we actually submitted disclosure for it while I was at Stanford. I'm not sure what the status of this is anymore. I think like after I won the faculty search, national search for a faculty position here, I think like we never actually completed the process. I will have to, ping them and ask them like, do they still want to like do this sort of stuff? They were like, you can actually make money on this, right? So think of it this way, like most people, when they build data commons, they have to store the data, right? They have to, for example, Encode DCC pays $35,000 per month to store their data. That's just something they have to do. And they have like huge Amazon grants that cover that data expense, but it's huge, right? $35K a month. Whereas here in HeartBio Portal, instead of storing, you're streaming. So like this, like it says on this slide, don't store it, stream it. Like why buy when you can rent? So it's sort of a new computing concept in a way that you're introducing. And whenever there's money involved, right? If you don't have to pay $35K a month, if you can pay much less than that, like why wouldn't you, right? So that's why hence, the disclosures here. The reviewers did not give us a hard time with this when we submitted the paper. Actually, they didn't say a word about this. So, but again, this was super cool, like computing stuff, right? So if you're interested in algorithms, and you're interested in, how can we, save money, right? Instead of storing, you stream. That's basically what this is about. This is a figure from the paper that we published, in April. It's pretty outdated now that we have so much additional data, but it sort of gives you an idea of the workflow. Also, this is a figure from the Cardioinformatics paper, sort of, maintaining HeartBio Portal by keeping up with new data. Momentum from databases like Geo is pretty hard, and it's also pretty important. So if you can automate as much of that as possible through these Internet-of-omics algorithms, right, can you actually like at least automate a portion of it so that the manual curation isn't as heavy? Like that is huge from a money-making perspective. So, that's a lot of fun. And also this was another figure in the Cardioinformatics paper that basically just showed where, at least at the time of publication of this paper, where and what kind of multi-omics data was found across different, for example, cohorts in TopMed, of which there are many, the All of Us research program cohort, the UK 10K project, the 100,000 Genomes project, et cetera. So it sort of gives you an outline of where data is with respect to cardioinformatics. I think that's it. Yeah, so so that covers all of the supplementary slides. I'm happy that that I got a chance to run through that quickly. So let me go ahead and see if there's any other questions here. Okay, great. Looks good. Thank you very much, everyone.
Thank you so much for this. This was great information. If you came in late, you didn't hear the chime, this is being recorded, so I'll be able to send the link out to everybody that attended the presentation tonight. Thank you for coming to hear the talk. Thank you, Dr. Homechuk, for giving this great presentation. And I hope everybody has a great evening. And whoever has class tonight, have fun in class.
Thank you.
Thank you.
Thank you, everyone.